Hi everyone! A very good morning to our lecturer, Dr. Norsazilawati Saad, and our fellow friends. Welcome to our presentation. We are from Group 3, PLP 3204, Fundamentals of Plant Pathology. Today, we are going to present about plant disease and its management. Here are my group members and their matrix number. Without further ado, let's get started. First of all, the plant disease that we choose is Papaya Ringspot disease or PRSV. This is a disease that caused by Papaya Ringspot virus from the Potivirus genus Potiviridae family. It has a host range of papaya and cucurbits such as cucumber, melons, pumpkin, squash, and zucchini. It has a wide distribution all over the world, such as Asia, Africa, North, South, and Central America, the Caribbean, Europe, and Oceania. PRSVP disease is also recorded from Guam, French Polynesia, and Northern Mariana Islands. Now, I will explain about the disease cycle of Papaya Ringspot virus. The disease cycle is categorized into a few stages, which are inoculation and penetration, infection, colonization, disease, dispersals, and survival stage. The first stage is inoculation and penetration. In this stage, aphids as the vector will feed on the diseased crops and carry the PRSV virus in their stellate. The virus will infect the susceptible host, which is the papaya tree in this case, by mechanical inoculation through the wound on trees. On the next stage, which is the infection stage, the aphids that feed on another healthy plant after feeding on infected plant will transmit PRSV virus into the healthy papaya plant. The virus viral nucleic acid will then free from the coat protein. While during the colonization stage, the virus viral nucleic acid will replicate in the host cells. Then, some of the virus move through plasmodesmata to adjacent cells and replicates. This virus affects various stages of plant growth, from seedling to fruiting stages. After that, the infected plants start showing the symptoms during disease stage. There are a few examples of symptoms such as the mosaic pattern, dark green blister, distortion and reduction of leaf lamina, dark green ring and oily streaks on stem, reduction in plant canopy and fruit size. In certain cases, the papaya trees will not produce yield due to the infection. Last but not least, after the disease stage, PRSV disease will undergo dispersal and survival stage. The virus will disperse through different species of aphids, especially the green peach aphids, that feed and transmit virus from diseased plants to healthy plants. The cycle will return to inoculation and penetration stage, and the cycles will go on. Now, I'm going to explain about the life cycle of Papaya Ringspot virus. For your information, PRSV is residue its whole life within the plant cell. First, it started with the vector, like aphids, feeding on the infected papaya, and then it will subsequently feeding on a healthy papaya. So this is how the virus spread from one infected plant to another healthy plant. So there is no incubation period for this disease to develop. Next, the virus will use the resources of the papaya tree for its own reproductions. After that, the virus will enter the new host plant. It will then shed its own protein coat and uses the host plant system to replicate its own genome or RNA. This is to make the protein coat for a new virus. The process is also known as replication. Next, it is followed by the virus uses the protein code to produce a new virus particles. After that, the new virus particles that have been produced will remain in the infected plant cell, and it will usually spread by aphids, and now we can see that the cycle will goes on and on. How to detect the papaya ring spot disease in farm? We can observe the symptoms, right? So here are some of the symptoms of papaya ring spot disease we can observe in our farm. Firstly, we can observe the tree leaf. When the papaya tree leaf develop mosaic and chlorosis on its leaf lamina, it could be a symptom of papaya ring spot disease. Besides the leaf lamina, we can also observe the petioles and the upper part of the tree trunk. If these parts have a dark green streaks, it could be another example of symptoms for papaya ring spot disease. Besides that, this papaya ring spot disease will also cause distortion of young leaf in papaya trees, especially when the trees are infected at the young stage. This will cause the trees remain stunted and affect its production. This leads us to the last point, which is the papaya fruits. When the papaya trees is infected with papaya ring spot disease, the fruits will have bump and ring spot. This will make the fruits unappealing and lose its market value. Besides observation with our eyes, we can also carry out diagnostic tests to detect the presence of papaya ring spot disease. Here are a list of diagnostic tests that is usually carried out in the lab. First, we have a dot immunobinding assay or DIBA, followed by immunocapture RT-PCR. After that, we have direct antigen coating DAC-ELISA diagnostic test. Also, we have SDS-PAGE diagnostic test, and followed by inclusion bodies test, and lastly, the electron microscopic diagnostic test to detect the presence of papaya ring spot virus in our farm. Meanwhile, for the disease management of PRSV, it can be carried out by quarantine prevention. This means that we should prevent the entry of infected papaya or prevent the importation of papaya from the country that has PRSV virus. It is our responsibility to protect the crops in our homeland from the disease infection, or else it will cause a huge economic loss in our agriculture sector. Next, PRSV disease can also be managed through biological control. For example, farmers should disinfect their working tools such as hoes, scissors, by dipping them in 0.525% sodium hypochlorite solution or Clorox, and then rinse them in water and use as usual. Other than using sodium hypochlorite solution, the working tools can also be disinfected in an oven at 150 degrees Celsius for an hour. These methods can prevent the spreading of virus from the tools to the farm. Besides that, another method of disease management is planting resistant crop varieties. Plant breeders have the responsibility to introduce the mild strain of PRSV virus into the host plant. These methods can protect the host plant from the more serious virus strains. We can say that these virus methods work like a vaccination that induce immunity or resistance in our host plant cells. Also, resistant varieties can be produced by genetic modification, such as the insertion of the virus coat protein gene into the papaya DNA. The next disease management for PRSV disease is cultural control. Through cultural control, farmers should restrict the movement of people within the plantation. This is also happening in our daily life currently due to the COVID-19 pandemic in our country. We name the lockdown as Movement Control Order or MCO. Both actions have the same objective, which is to prevent the spreading of virus. In papaya plantation, aphids, which are the disease vector, could be taken into plantations on people's clothes, tools, vehicles, or machineries. Other than that, diseased papaya trees should be removed as soon as possible. Same goes to the severely affected blocks of papaya trees. All of these should be eliminated to cut off the virus and prevent aphids from spreading the virus into other farms. Last but not least, farmers should grow papaya trees under netting. However, this method is very expensive and not suitable for small-scale producers or subsistence growers. On the other hand, PRSV can be controlled through chemical methods. Unfortunately, there is no chemical treatment for viral infections. Hence, insecticides can be used to kill aphids or the disease vectors. Overall, PRSV is an aphids-borne disease which are non-persistent. This is because the aphids can only transmit the virus particles into a healthy plant for once. The virus particles will not stay too long in their stylets. Hence, farmers should reduce the initial inoculum or primary infection of virus by destroying infected plants or debris. As saying goes, prevention is better than cure. Farmers will be able to reduce disease incidence by reducing the initial inoculum of PRSV virus. So, that's all from Group 3. I hope you enjoyed the presentation and thank you for paying your attention. Have a nice day.